Good morning. So welcome to our application lab. I'm here with Maria, and today we will show you cell isolation, how to prepare your plate and your samples, and how to perform the experiment. So Maria will take the camera. So what you need for this experiment, today we will isolate CHO cells in 12-well plates. So you need the CHO cells here that I already put with L15 medium, which does not contain FBS. So it does not inhibit the trypsin 10X here, that we will use to detach, to dissociate the cells, and that we will use to load the FluidFM micropipette that we have here. A FluidFM with an 8 micron micrometer aperture size to be able to pick up the cells. The CHO cells are around 10 to 15 micrometer. Before performing the experiment, we will need to coat this micropipette with a solution, the Pacram, which is an anti-fouling reagent that I already filled into this well here. And I prepared also 3 wells of PBS to wash the micropipette from the Pacram, wash the Pacram from the micropipette. So I will place this 12-well plate that contain both the cells and the Pacram solution into the bot system. We'll put it here on the left port of the bot system and remove the lid of the 12-well plate that I will keep inside. And now I will prepare the micropipette. So that comes in this blister package here. I will gently open it, take it out, and I will load it with 1 microliter of trypsin 10x concentrated, as when we will deliver it into the medium, it will diffuse a lot, so it will decrease the concentration. That's why we use 10x concentrated trypsin. So I will load the reservoir of the FluidFM probe with 1 microliter. Click a little bit in case there is any air bubbles, and place it here into the probe holding plate. We have 4 positions, or we can put the probe. And now I will put this probe holding plate into the right port of the bot system. And now we can switch directly to the software to configure the system and prepare the probe. So here we have on the left 12-well plate with content 8 at 300 micrometer, and on the right the loading plate. So first, as we are using a new FluidFM probe, we will prepare the system here, and we will drop the old probe, which is already mounted on the system. It's a probe that we used previously for another experiment. We'll drop it in an empty position here and pick the new ones that we have placed in A1. So here it's moving, it's released the old probe and moved to A1 now here. I will switch back to the 10x objective here and focus on the FluidFM micropipette. To finalize the gripping, I need to center it here and click on OK, and the head will go down to finalize the picking. And we will see probably a little offset after the gripping. Yes. And during the gripping, after the gripping, there is a tightness measurement here to make sure that there is no leak and that the gripping has been done properly. Then we will switch directly to fill the probe here. I will switch to the 20x objective. It's a bit easier to see usually. Okay. I click here on set new reference. This is a little help image here to make sure that we have filled the nanosyringe. And I will start to use maybe 500 millibar to fill. Click on fill here and wait a few seconds and maybe increase the pressure if it's not enough. Here it has been filled. We can see it comes a bit out of through the aperture here. I go back to idle pressure and I will go directly to the Pacram solution here that is in C1 to start the coating as fast as I can. The coating will last 30 minutes. So it's right now 10:51 here in Zurich. So we will wait half an hour for the coating. In the meantime, what we can do here is finalize the prepare system workflow. So I will align the laser, a step that we skipped previously. For that, I need to remove the shutter from on the microscope, which is hiding the laser from the camera. It's for and click on align here. Here we have it, and I need to align this laser on top of the micropipette, and especially on top of this dark shape here, which is a reflective coating, the gold reflecting layer of the micropipette. So we need this laser, I remind you, for the force feedback control that gives to the system information about the position of the micropipette. Now that I aligned it, I can put back the shutter here, click on OK, and we can now maximize the signal. What does it mean? Inside of the head, we have some mirrors that reflect the laser onto the photoreceptor, and we need to get the best position of the laser on the photoreceptor, and there is an automatic system that controls that to get the best signal from the laser. So here we have a very nice signal. Okay. Configure the probe. You can scan the blister package that I showed you before. There is a QR code on it that I just scan it, and it gives us some information about this micropipette, and especially an SCM picture of the micropipette that we are using right now. We take a picture of every single probe that we provide and give every information about them for the customer for the experiment. Okay, here. The spring constant, it gives information about the stiffness of the probe. It's required when you want to measure precise adhesion forces, for example. It's not the case here in isolation experiments, so we will skip this step. We don't need to do that. Filling the probe, we did it before. Why did we do that before? Because we need to fill it before we dip the probe into liquid. Otherwise, you have air inside, and it's impossible to push air through the liquid with the pressure controller that we have, with the 1 bar pressure that we can apply, which is the maximum. So we do that before. Go to the sample, here we are. We don't need to re-maximize the signal again. It's only needed in case if we do that in air before and then in liquid, and in that case, the reflection of the laser is different. Escape. And sensitivity, the sensitivity depends on where you align the laser, but also depends on the reflective gold layer. It's like the spring constant, only required if you need to measure precise forces. So we will skip that part. And finally, we need to align the probe. I will do it here, but I will redo it anyway later once we are in the medium with the cells to have the most precise alignment. What is it used for? It's to give the software the information to where is the opening of your probe. So when you target a cell, it goes to this exact position and not somewhere else. Okay. And now we have to wait another 25 minutes. We can switch back to the camera. We will just show you because right now we have the 12 well plate where we have the cells, but we need also a 12 well plate with medium where we will isolate the cells. And this 12 well plate, we already prepared it actually with L15 medium, also without serum. Why? Because the serum will make a kind of coating at the bottom of the plate, and it's easier to release the cell if you use a medium without serum. So we already prepared actually a 12 well plate here with the medium. So it's the same well plate than before, and that we will place into the bot system at the instead of the probe holding plate. So I will go quickly to the software and use the exchange plate workflow to ask to the system to go to the right port, and I will put the plate in the right port. Now we can go back to the software and wait until the Pacram coating is finished. I will bring, when I change the plate, it brings the probe out of the well. I will go back quickly. The coating is still going on when I'm out of the well because there is always a droplet around the micropipette when we remove it from the liquid. So that was continuing the coating. Now I bring it back. I'm out of focus, but it's here. And now we can just wait until the coating is done. So it started at 10:51, so we still have more than 20 minutes to wait, and we will come back. So it has now been half an hour that it is coating, so it is finished now, and we will rinse the probe into a PBS here, which are in C2, C3, and C4, by just using the navigation tool to dip the micropipette a few seconds in each well. Secondly. The first well, the second well. On the right here, we have just before we have placed a 12 well plate here. I forgot to change here in the exchange plate workflow. You can put that we have now a 12 well plate here as well. Apply. Now I finish the rinsing. And we can go now to the cells. For example, in B2, we have cells there. Okay. And I will approach the surface here using the probe control tool here. In the setting, you can control the board settings. Here's the approach speed. I will increase it to 50 microns per second. We'll arrive a bit further. For later on, I will change also the retraction speed to 4 microns per second, and the retraction, safety retraction to 2 microns. Okay, I retract again by 10 microns, and using the pressure control tool here, we will apply some pressure to start to release the trypsin around these cells here that we want to detach, and wait a few minutes that the trypsin dissociate the cells. You can play with the pressure depending on how many cells you want to detach. If you want to detach only a very specific cell, you can approach to it, apply only 50 millibar or so on. Or here we want to isolate 12 cells, so I will apply like 500 millibar to dissociate a lot of cells around this area. You can move a bit around to deliver the trypsin a bit everywhere. So here, obviously, the trypsin is not fluorescent, so we cannot see it. But if you mix it with some fluorescent compound, you could be able to see exactly the diffusion of it. So depending on the cells, their confluency, it might take more time or less time to detach them. What we recommend is to seed the cells the day before, and in order to obtain a, let's say, 50% confluency, so a bit less than what we have here, but it should be okay still. We will start already to react. We will apply 1 bar pressure maybe, and wait a bit. We'll start to here detach a little bit. It's a bit slow, but moving a bit around. So applying pressure. Here as well. You can follow live here the pressure which is currently applied. This one starts to detach a little bit. We'll approach again to be a bit closer. Here we see a little movement. So when they are highly confluent, it's more difficult, of course, to detach them like that. But here we can see that, yeah, they start to finally detach. This one we can maybe try to pick up. So we will use the adhesion workflow here. I will retract a little bit. I need to recenter. I'm close to the surface. I need to recenter the probe here. Okay, so when I will target, I pick up the good cells. Here we see they finally start to detach nicely. So I will target this one. I will stop to release the pressure here, go back to idle pressure. Okay, and use the adhesion workflow here. So we will use a 70 millivolt set point. This is the force that the system will measure to know that it has touched the cells. We will keep an approach movement speed of 50 microns per second. The pause is the duration of the pressure that we will apply, and we will start with minus 50 millibar, check if this is enough to pick up the cells, and with a retraction distance of 50 micrometer. We will click on measure here. We'll see if it's enough already. Not enough, so probably we need to reapply a bit more here, more trypsin. Or what we can do is try to apply a stronger negative pressure here. For example, minus 80 millibar. Almost. Probably it's not yet enough detached, as we have seen it takes some time. It's also connected still to the next cells. Probably that's why it's a bit more tricky to pick up. Let's try this other one here. Maybe minus 80 millibar is not enough, simply not enough to pick it up. We can also increase the set point, so it's sometimes help a little bit. And as soon as it has picked up the cells here, I go back to idle pressure here, and we can go to the move to the next well where we will depose the cell. So usually a simple approach is enough to depose the cells, but it can happen with the first 2, 3, 4 cells that you have to move a little bit and apply some pressure. I will apply 50 millibar pressure and then approach to the surface here. I've touched the surface. Then I can try to retract a little bit. I see that the cell is still around, probably, and try to move away. So we can see it's still a little bit attached to the cells. We will try to play a bit around with it. Wait a little bit. Maybe it helps the cells to adhere. And here it is. It has been released. Now we can use it's quite common that it happens like that for the 2, 3, 4 cell, we will see. And then it should be more straightforward, release the cells again and again, like really easily. So we can use this history tool here to go back to the position where we have when we have started to dissociate the cells already. So we don't need to wait another 5 minutes to pick up a new cells. Here, I will focus on the cells here. Use adhesion workflow again. Maybe I can reapproach and reapply some pressure to help to associate a bit more the cells around. But they start already to detach all of them, we can see, compared to before. So we'll try to pick up a next one, maybe this one here. We will try to use the same parameter. I was probably a bit not fully centered here on the cells. That's why with this other one here. We need to those are still not fully. I will apply a bit more trypsin here again. Seems a bit hard to detach today. So we'll go back to idle pressure. I get this one. I don't want to apply stronger under pressure than minus 100 millibar. It might be then more difficult to release the cells. So I prefer to, no. So almost come. What we can try is maybe a slower here approach and movement speed. So when we retract, it does not move too fast and leave the cells on the surface. And here it goes. And we can move to the next well here. A2. Apply again 50 millibar and approach the surface. Just a pulse of 50 millibar and we will see if it's enough this time. I can retract a little bit and see if it's enough. Still not enough, so I will apply a bit more pressure. Wait a little bit here. Back to idle. And here it is. We use again the history tool here. Move again there. And apply a bit pulse of trypsin in case there is any debris stick to the micropipette. So I like to apply this pulse of 1 bar pressure from time to time in between cells. Reapproach again here. So I will keep a bit of pressure to continue my trypsinization of the cells all around and target the next cells here, for example. So I keep the same parameters on the previous cells, and let's see. Not enough. This one. But they're touching each other, so it's not the easiest. Let's try this one. Can go here. This one seems a bit more isolated. They stick really well to the surface. Probably we need to wait a bit. The trypsin. What we can do is increase the time also to 7 seconds here. Actually, this one is a bit. So I continue to apply some pressure here, and we'll try maybe if another one is already okay. Let's try minus 150, but not much more. Here we have this one. Let's go to C to A3. Wait a little bit. We'll retract. Still the same here. It's only the third cell that we are isolating. Wait a little bit here. Can try to retract from the liquid and goes in. And here it is. Try to pick up this one. Apply again a pulse of 1 bar. I will lower down here to minus 100 millibar because probably we got some issue to release it due to this strong under pressure when we picked the cells. So you have to find a good balance between enough under pressure to pick it up, but not too much, otherwise it's getting tricky to release it. With big cells, but yeah, we could pick it up. And let's move to A4. Apply 50 millibar again and approach the surface. What we can try is a retraction of only a few microns. 5 times and move away. And now the cell has been released. So now it's getting easier and easier. That's quite common that after 3 or 4 cells. But let's see with the next one, and then we will finish to isolate the 12 cells here. So to pick up this one, I apply again a pulse of 1 bar in case there is any debris from the previous cells. It's always nice to do. You can also do during the movement of the micropipette from in between the wells. It's approaching now a bit slower as we change the parameters. But let's see now. And we got it. Now we can go to B1. Here. Apply 50 millibar and approach the surface. Now retract and move away. And the cell is here. We can go back again to the trypsinized cells. And target another one. Don't forget to apply this pulse of 1 bar. It really helps both for the detachment and for the picking. And let's keep the same parameters as so far this seems to be good one. Let's try this one. I think we already tried previously this one. If now it is detached enough, let's see. Also, when you have a lot of cells around, it might, impair with the set points that we have set up. So that's why here probably we are not touching it because we have touched these cells on the side, and that kind of artificially modifies the set point. Let's see. We got that one. Let's go to B2. If you have another cell that is below the aperture, like touching the micropipette, it bends over the micropipette and moves the laser, which give a fake signals and then a wrong set point. Apply the 50 millibar and approach. Let's retract. And we have it. And move to the next one. Can apply a pulse of 1 bar here around, and let's target, for example, this one here. This is one of the cells we already tried before. Let's see if we are more lucky this time. Yes, and we can move to B3. So these are 7 cells that we're isolating. It's now getting faster and faster, obviously. So it's quite common that it's like that when we isolate the CHO cells. Don't forget to apply this 50 millibar pressure. It really helps the release of the cells here. Touch the surface. I'm always focusing so you can see nicely, but it's not really necessary. Actually, you can do it blindly. And we move, and the cell is released. So we can move again back to where we trypsinize the cells. Apply this pulse of 1 bar pressure. And try to pick up this one that we previously failed because there was the other one on the way. And let's try if this time we're a bit more lucky. Now we remove the cells that were interfering with micropipette. And here we have it. Okay, we can go to B4. So I will apply 50 millibar and approach against the surface. I'm already on the surface. I can retract and move away. And then you can move again like that, faster and faster. Of course, I take time here so you can see it nicely. So we can go back here. So you see now like a lot of cells have detached around. The trypsin has been taking effect. We can try to pick up this one. It's not fully detached, but I think it's should be okay. We can try at least. Don't forget to apply this 1 bar pulse here. It's always nice for a few seconds. Then we can approach. Taking of the cells now. Yes. Go back to idle and move to C1. Apply the 50 millibar here and approach the surface. Retract by 5 micron and move around and go. It's here. Let's try to pick up this one. I think it's one we tried before. Don't forget this 1 bar pulse. I was about to to forget, but if you want a really nice pickup and then measure. I'm not sure that we can still see some attachment here, but let's see if the parameters are strong enough to pick it. Okay, now I'll go back to idle and I can go to C2 again. So if you want to remember where you were before, you always can go in the history here, and here you see I was in C1 before. Here I have also Maria who helped me to to remember, so that way I don't check all the time. But that's something you can do like that. Approach the surface here, retract by 5 micron, and move up. This time, sometimes it happens from time to time. We will wait a little bit maybe. And here it is. It might help sometimes to wait just a few seconds. I will apply this 1 bar pulse, like during the movement here. There is always some liquid around the micropipette, so it will stay a bit around. And probably the trypsin also help to remove any debris. So we will now pick up another cell. We can try this one here. I will release this 1 bar pulse with this 1 bar pulse and go with the same parameters again. Maybe that the cells are nicely detached. I could actually use minus 50 millibar instead of minus 100, but let's see. Not this one. You see that we see was still a bit attached. Let's reapply a pulse here. After a while, they start also to readhere once the trypsin has moved away. So I reapply a bit of trypsin here. Let's try maybe this one. No. Probably they start already to reattach, or we have, let me check the micropipette. No, I don't see any debris. It's not always easy to see. Keep the same parameter. I don't want to play too much with it. Okay, and we're going to go to C3 now. So from time to time, it's nice to re-shower the cells a bit with trypsin. As after a few minutes, they start to reattach. That's normal. Cells, they take 5-10 minutes to start the reattachment. Now we approach the surface again. Focus. I retract a little bit. And we can move away. And we can go back and to pick up the last cells. We can try this one here. I will apply a bit this pulse of 1 bar pressure here for a few seconds. Keep the same parameters. So this noise that you might hear is when the pressure controller is releasing the pressure. We hear it every time we pick up a cell or when I release a bar.
It's totally normal.
The big cells, I hope the parameters are strong enough to pick it up, but let's see. Yeah, almost. Maybe we can try another one, like that could be easier, or we need also to show a bit more, like with trypsin. because I've been like quite along now without showing everywhere.
And also trypsin gets diluted with the medium with the time, so that's why it's good to release from time to time trypsin over the cells when isolating.
I retract a bit the nanosemi, the micropipette before we pick it up. Yes, we got it. And let's go to the last well here. Apply 50 millibar here. Approach. Remove that and retract. 5 microns. Ah. Wait a little bit. Go back to idle. Move. And now we finished to isolate the 12 cells. So now what we will do is, of course, wait maybe 20-30 minutes, or even maybe more. Like it's safer to wait like at least 1 hour before we replace the medium, so it gives time to the cells to nicely attach, and then we will replace them with a complete growth medium, DMEMF12 in the case of the CHO cells with serum, and replace it in the incubator to check on the next day. And from our experience, 95% of the cells will give a new colony.
And then probably these colonies would be more visible after 2-3 days, because then the next day you can just try to look for your cell isolated in your well, but
sometimes they're already divided, but sometimes you have to wait a bit more.
That's it.
But they will be at least nicely attached. We will see that on the next day. And after maybe a week, a little bit more, you can split them again and have a nice new monoclonal culture. Okay. So thank you for your attention, and I hope this will be helpful for you in your experience.
Thanks.
Bye.